Smell or olfaction is a visceral sense because of their close association with gastrointestinal function. Smell and taste receptors are chemoreceptors that are stimulated by molecules in solution in mucus in the nose and saliva in the mouth. Because stimuli arrive from an external source, they are also classified as exteroreceptors. The sensations of smell and taste allow individuals to distinguish between estimates of up to 30 million compounds. Olfactory sensory neurons are located in a specialized portion of the nasal mucosa, the yellowish pigmented olfactory epithelium. In humans, it covers an area of 10 centimeters square in the roof of the nasal cavity near the septum. The olfactory epithelium is said to be the place in the body where the nervous system is closest to the external world. The human olfactory epithelium contains about 50 million bipolar olfactory sensory neurons interspersed with glia-like supporting or sustentacular cells and basal stem cells. The olfactory epithelium is covered by a thin layer of mucus secreted by the supporting cells and Bowman glands, which lie beneath the epithelium. Each olfactory sensory neuron terminates in a knob containing 6 to 12 cilia. The axons of the olfactory sensory neurons enter the olfactory bulbs. In the olfactory bulbs, the axons of the olfactory sensory neurons contact the primary dendrites of the mitral cells and tufted cells to form olfactory glomeruli. The olfactory bulbs also contain periglomerular cells, which are inhibitory neurons, and granule cells, which have no axons and make reciprocal synapses. At these synapses, the mitral or tufted cells excite the granule cell by releasing glutamate, and the granule cells in turn inhibit the mitral or tufted cell by releasing GABA. Free endings of many trigeminal pain fibers are found in the olfactory epithelium, which are stimulated by irritating substances that leads to a characteristic odor, such as peppermint. The axons of the mitral and tufted cells pass posteriorly through the lateral olfactory stria to terminate in 5 regions of the olfactory cortex: the anterior olfactory nucleus, olfactory tubercle, piriform cortex, amygdala, and entorhinal cortex. From these regions, information travels directly to the frontal cortex or via the thalamus to the orbitofrontal cortex. Conscious discrimination of odors is dependent on the pathway to the orbitofrontal cortex. The orbitofrontal activation is generally greater on the right side than the left. The pathway to the amygdala is probably involved with the emotional responses to olfactory stimuli, and the pathway to the entorhinal cortex is concerned with olfactory memories. The nasal cavity contains another patch of olfactory epithelium located along the nasal septum in a well-developed vomeronasal organ. This structure is concerned with the perception of odors that act as pheromones. Vomeronasal sensory neurons project to the accessory olfactory bulb, and from there to the amygdala and hypothalamus that are concerned with reproduction and ingestive behavior. The organ is not well developed in humans. The sense of smell is said to be more acute in women than men, and in women it is most acute at the time of ovulation. Smell has a unique ability to trigger long-term memories, a fact noted by novelists and documented by experimental psychologists.